Hello everyone, hope you're doing well during these difficult times. Whether you're a medical student, a pre-med, or simply someone that is curious enough about learning how energy is managed in your body, understanding the fundamentals of metabolism is pivotal for your success in both biology and biochemistry. Not surprisingly, these subjects are covered extensively on both the IMAT and the MCAT. Therefore, it is important to familiarize yourself with the main metabolic pathways that take place within the cell. So why not begin with these two? Am I right? In this opportunity, I have decided to omit glycolysis because it is not essential for our understanding of the mechanisms that we will go over today. Instead, consider the intermediate step between this pathway and the Krebs cycle. Your professors may also opt to call it the Tricarboxylic acid cycle. In order for the pyruvate that was produced from glycolysis to enter this pathway, it must first go through a series of modifications that will culminate in the synthesis of a special molecule known as acetyl coenzyme A. The product of this intermediate step is then able to incorporate coenzyme A into the cycle so that it can be oxidized to produce energy for the cell. You should take note of the chemical reactions that take place during the course of the DOTRA step, as I like to call it, an acronym that stands for the three fundamental reactions that we are observing: decarboxylation first, oxidation second, and transfer of an acetyl group to coenzyme A lastly. Notice as well that each atom on the image is color-coded. So red stands for oxygen, black stands for carbon, blue stands for sulfur, and the hollow circles represent hydrogen atoms. The small pair of yellow icons are supposed to be electrons leaving the acetyl group. And then there's the orange box that is supposed to be the coenzyme A portion. Having undergone the modifications pertinent to the DOTRA step, pyruvate is able to access the Krebs cycle as acetyl coenzyme A. During your biochemistry studies, take note of where these pathways take place within the body. For example, within which organelle and region. In this case, all of the reactions outlined on the picture occur within the mitochondrial matrix. Pyruvate dehydrogenase catalyzes the conversion of pyruvate to acetyl coenzyme A through the mechanism described on the prior image. Once the coenzyme A moiety is removed from the molecule, a 6-carbon compound is synthesized bearing the name citrate. Dehydration, namely the loss of a water molecule, or dehydration of citrate in this case, is going to lead to the emergence of a double bond in the structure. Remember that when bound, carbon atoms must be surrounded by 4 electron groups at all times, which is then hydrated, and I'm of course talking about the double bond here. So the double bond is hydrated to create D-isocitrate. The cycle then more or less follows this reaction pattern to concurrently reduce NAD to NADH and FAD to FADH2. These are two fundamental electron carriers whose importance will be touched upon next. Also take note of the intermediate step that is catalyzed by succinic dehydrogenase, because the coenzyme product, ubiquinone, which shows up as Q in the diagram and then reduced to QH2, has long-term implications for the metabolism of the cell. In biochemistry, and nearly all sciences with a biological foundation, it is imperative to comprehend how the smaller, less relevant processes or procedures affect the rest of the body. In the case of metabolism, all of the pathways that you will study are intricately connected by a series of reactants and products, which meaning that the products of one pathway behave as the reactants for another, hence maintaining the functionality of the biochemical machinery that is necessary to perpetuate life. If we use the breakdown of glucose, also known as glycolysis, as a starting point in this case, we observe that the neo-synthesized pyruvate molecules serve as the base for the production of acetyl coenzyme A, which can then enter the Krebs cycle and contribute the reduction of NAD and FAD to form NADH and FADH2, as I mentioned before. Once reduced, both of these molecules can serve as vehicles for the potential energy contained within these electrons, which subsequently are exploited by the ensuing pathway to generate high quantities of energy for the cell. We are talking, of course, about the electron transport chain. Since all of the components pertinent to this process are located deep within the mitochondria, all of the metabolic processes pertaining to this pathway occur inside of this organelle, of course. Embedded on the inner mitochondrial membrane, we find 5 interesting characters whose functions and roles in the electron transport chain are much more fundamental than their names, which is why they're listed as complex 1 through 4 in this schematic. Protons are pumped out of the mitochondrial matrix and into the intermembrane space as a consequence of electron flow through the major components of the ETC. This creates a concentration gradient of hydrogen ions through which ATP synthase can do its job. Additionally, the ATP contribution differs between the 2 electron carriers, with NADH electrons enabling for the synthesis of an additional ATP molecule, when rounded up, of course. Notice that I included both the exact and rounded up yields because the time will come when you will have to compute these values into a sophisticated stoichiometry problem, as an example. So it is to your advantage to familiarize yourself with these numbers as you advance your course. At the moment, it is not necessary for you to understand precisely how the ATP synthase does its job. But if you need help with this enzyme, feel free to reach out, because I'm here to help. Remember, lastly, that biochemistry is a rather complicated subject which seems to contradict itself at times. It is important, thus, that you remain patient, calm, focused, and passionate if you want to succeed in your learning and ultimately do well in class. Pay attention to the details, but always focus on the bigger picture, as I mentioned before. Feel free, of course, to leave your questions in the comment section down below, and I will tend to them at my earliest convenience. Thank you for watching, and hopefully this tutorial was a helpful summary of this whole mess. Good luck!